circular pieces of DNA that exist outside of a bacterium's main chromosome. In genetic engineering, scientists use them like tiny delivery trucks to carry new genes into bacteria. For example, we can insert the human insulin gene into a plasmid, place it inside a bacterium, and let the bacteria produce insulin for us. This is how companies like Genentech created synthetic insulin for diabetic patients. Plasmids are naturally absorbed by some bacteria, which makes them ideal for transformation in the lab. Mini summary: plasmids are circular DNA used to insert genes into bacteria. They're natural, easy to manipulate, and crucial for biotech. Quick quiz: what are plasmids? Circular DNA molecules used to transfer genes into bacteria. What's an example of plasmid use? Producing human insulin in bacteria. Section 2: restriction enzymes. Now let's talk about restriction enzymes. These special enzymes act like molecular scissors. They cut DNA at specific sequences. One of the most famous enzymes is EcoR1, discovered in E. coli. By cutting DNA precisely, scientists can control what genes go where on a plasmid. Most plasmids have maps showing where restriction enzymes can cut, which helps us insert genes exactly where we want them. Mini summary: restriction enzymes are tools that cut DNA at known spots, giving us precision in genetic engineering. Quick quiz: what do restriction enzymes do? Cut DNA at specific sequences. Where was EcoR1 discovered? In E. coli. Section 3: genetic engineering in eukaryotes. Eukaryotes are more complicated. Unlike bacteria, they don't have plasmids. So how do we genetically modify them? By using viruses. These viruses act as delivery systems, injecting DNA into cells. This is how animals like glowfish are made, by inserting fluorescent genes into their DNA. Scientists can do this with precision, even though public opinion on GMOs is still mixed. Mini summary: we use viruses to genetically modify eukaryotes since plasmids don't work on them. Quick quiz: why can't we use plasmids in eukaryotes? Eukaryotes don't naturally absorb plasmids. What do we use instead? Viruses that inject DNA. Section 4: electrophoresis. Electrophoresis is a lab method used to separate DNA fragments by size. DNA is negatively charged, so when we apply an electric current, DNA moves toward the positive electrode. We run it through a gel, usually agarose, which slows it down. Smaller fragments move faster and farther. This helps scientists identify DNA sizes and patterns. Mini summary: electrophoresis separates DNA by size using electricity and agarose gel. Why does DNA move in electrophoresis? It's negatively charged. What does the agarose gel do? It slows DNA movement, separating it by size. Section 5: PCR, polymerase chain reaction. PCR is like a DNA copy machine. Invented by Kary Mullis, PCR lets us amplify tiny amounts of DNA millions of times in just a few hours. It's used in crime scenes, COVID-19 testing, and more. You need a special machine, a DNA template, primers, and enzymes. It's fast, accurate, and essential in diagnostics. Mini summary: PCR quickly copies DNA millions of times, making it essential for labs and medical testing. Quick quiz: who invented PCR? Kary Mullis. What is PCR used for? Amplifying DNA for tests like COVID-19 or crime scenes. Section 6: DNA probes. DNA probes are short DNA strands with a marker on them, like a fluorescent tag. They stick to a specific DNA sequence. If that sequence is in your sample, the probe lights up. It's like a targeted search tool. DNA probes are fast and useful for identifying bacteria or viruses in medical diagnostics. Mini summary: DNA probes light up specific DNA sequences, making detection fast and accurate. Quick quiz: what are DNA probes? Short DNA strands with a marker that bind to target sequences. What are they used for? Identifying specific microbes or genes. Section 7: binary fission and bacterial growth. Now let's shift to bacterial growth. Bacteria reproduce through binary fission. One cell splits into two. They don't go through mitosis like humans. Instead, they copy their circular chromosome, stretch out, and split. Under ideal conditions, this can happen every 20 minutes. That's why infections spread fast. Bacteria follow 4 growth phases. 1, lag phase, getting ready. 2, log phase. Rapid growth. 3, stationary phase, resources run low. 4, death phase, cells start dying off. Mini summary: bacteria grow fast through binary fission and go through 4 growth phases. Quick quiz. What is binary fission? Asexual reproduction where one bacterium splits into two. Name the 4 phases of bacterial growth: lag, log, stationary, and death. Hasha Section 8. Normal flora and microbiome. Let's finish with the human microbiome. Normal flora are the microbes that live on your skin, mouth, gut, and more. They help digest food, produce vitamins, and protect against harmful bacteria. This relationship is called Schalled mutualism. Sometimes it's commensalism. One benefits, the other isn't affected. Other times it's parasitism, where the microbe harms the host. When antibiotics wipe out good bacteria, it can cause problems. That's why balance is key. Mini summary. Your body hosts helpful microbes, normal flora, that support digestion and immunity. Quick quiz. What are normal flora? Microbes that naturally live on or in your body. What's mutualism? Both the host and microbe benefit. Final summary. Plasmids help us genetically engineer bacteria. Restriction enzymes cut DNA at specific sites. Viruses are used for gene transfer in eukaryotes. Electrophoresis separates DNA by size. PCR copies DNA for testing. DNA probes help detect specific genes. Binary fission drives rapid bacterial growth. Normal flora support your health. Let me know if you want a downloadable version, voice recording, or just the script split into